Hello, this is Blue Sci, the Science Media Society of the University of Cambridge, and we're going around the university, going to different science departments, trying to talk to different researchers and group leaders to get a better understanding of what's happening behind these walls. In this video, we'll be talking to Dr. Matthias Landgraf, who is a principal investigator at the Department of Zoology, to try to talk about how they use the simple system of the fruit fly in order to better understand nervous system development. Could you tell us a little bit about what the lab does?
Yes, so my lab's working on, we're interested in how nervous systems form, how brains develop, and so coming from that, we've decided we work with Drosophila because it's a simple model organism that we can work with identified nerve cells with, and in particular, looking at development, we start at the beginning. So we look in the embryo, and more we look at when this system, the nervous system, is finished. So that's the end of embryogenesis, and out comes a maggot which crawls around, which is really just a sort of grown-up embryo that has learned how to move around, find its own food, and sort of self-catering kind of situation. And that's what we look at to find out how does nature put these cells together so that it works.
How many neurons are in the larval nervous system?
So the number that most people like to use is about 10,000 nerve cells in the nervous system,
That's quite a lot.
Yes, it's a lot, but it's a tiny fraction compared to what one finds in let's say a mouse nervous system or the human nervous system. So it's that sort of intermediate level of complexity that makes it attractive to us.
Okay, so what kind of techniques do you use to study these larvae?
So we use primarily genetics.
So here are two males. This one has straight wings, which is how normal flies have their wings, and this one has curly wings. So we use what we call a genetic marker in order to label different chromosomes of the DNA, and one of those causes the wings of a fly to be curly.
And then we use that to mark them so we can visualize them, and then also to manipulate them. And then we use microscopy to look at these cells and how they behave, how the cells are interacting with one another, and we use behavior analysis to find out when we've messed them up, does it mean anything to the animal? Is there a behavioral outcome? Is the nervous system functioning differently? And does it allow us to understand how it's actually working normally? And we use electrophysiology to listen in to how these nerve cells talk to one another.
Could you tell us a bit about maybe a recent project in the lab and kind of what the science behind that has been?
Yeah, so what we I guess the projects I'm most excited about are they involve two bits. One is reactive oxygen species, which are sort of molecules normally thought of as dangerous and destructive that accumulate when we get old and are associated with neurodegeneration. And we wanted to find out what do they normally do. And we see that actually we might have to completely change of how we look at these molecules, namely as signals that are critically important for allowing nerve cells to adjust themselves depending on their circumstances, their activation level. So regulators rather than destroyers of tissues. And linked in with that, we're now looking at how these molecules might be used to communicate as signals between cells, between nerve cells and between nerve cells and glial cells. And I'm really excited about that because it's sort of digging up old stuff but looking at it from a different angle, and I think that's always exciting.
Okay, brilliant. Thank you very much.
Pleasure.